I think. Is the order the same as the? Am I?
It's I think the last is.
What does the order say?
It says Danny, and then Danny, Mike, John.
So you want to go with that order? No, I don't mind. Okay.
Is that good? Are we all set? Do you want to go next?
Don't make a difference. Mine. No, it's alright. I'll go second.
You sure?
Can I go last?
Yeah.
Okay.
No, John. Jonathan.
John.
Can you go last?
No.
Sorry, can we go last?
You want to go second?
Okay, fine. You go second.
Doesn't matter.
Okay. Let me start.
Yeah, right. Good afternoon, everybody. All right, so guys, welcome to Books and Bagels. I'm glad you guys all came out. My name is Jessica Snyder, and I'm the Vice President of the Graduate Student Association and the Graduate Student Association and the Office of Graduate Studies. I'm glad you made it out for the event. Thank you. Today's event is going to go as follows. We're going to have 3 graduate students talk for about 10 to 15 minutes about their research projects and the opportunities they've had in graduate school. And then we're going to have a moderated discussion where you guys get to ask questions, and we'll have just kind of a general discussion about interdisciplinary research and the projects you've worked on today. I'd like to introduce our moderator today, Ms. Dr. Anandan. She is an associate professor of biology.
Good afternoon, everybody. I am really delighted to be the moderator of today's conversations in interdisciplinary research. And we have 3 really fabulous presenters who are your peers and who will start to talk to you about why interdisciplinary research is such a good thing, some of the pitfalls, some of the positives. And the panel comprises of Dannielle Figueroa, Michael Ryan, and Jonathan Soffer. Dannielle is from the School of Biomedical Engineering, Science and Health Systems. Michael is from the Civil Architectural and Environmental Engineering Department, and Jonathan comes from the Department of Chemistry. They're all PhD candidates, which means they crossed that awesome hurdle of candidacy, and are here to tell you about their work. So, Dannielle. By the way, we would appreciate it if you could hold your questions for the panel until the end when we do the moderated discussion. This way you'll focus on all 3 of them, see what they have to tell you, and then we'll open it up for questions later. Thank you.
So, thank you. So my name is Dannielle Figueroa, like she said. I am in biomedical, but I work in mechanical engineering, like so many biomedical students, which makes this very interdisciplinary appropriate. And hopefully by the end of the talk, you guys will grasp at least some knowledge of like diabetes and why it's bad for you from a mechanical standpoint. So just to give you a little background about myself. So I did biomedical engineering before in undergrad as well. I went to Tulane. Tulane's a very research-driven school. Like Drexel's very into co-ops. Tulane is very like research-intensive the whole time. So everything I did there was all like orthopedic, bones, mechanics, breaking things with my hands. And then I worked for a short time at the VA in Texas, which is also joined with the Health Science Center there, where my primary focus was then on muscle regeneration. Mainly what the target is, San Antonio has like 4 military bases, so it's very, very military driven. There's a lot of invitations, a lot of things like that. So that's mainly what drives their research. And then when I came to Drexel, I've gotten into cell mechanics research. So kind of gone smaller and smaller and smaller, and hopefully when there's a postdoc, I won't be going any more smaller. I mean, it clearly looks like proteins are in my future because, I mean, where can I go from there? And I don't actually know where I'm going yet. I know I'm finishing. I'm just now focusing on where I'm going, so it's all up in the air. So like I said, my lab mainly focuses in on the effects of diabetic hyperglycemia. So just to give you guys a little background about what diabetes is. I'm pretty sure most of us in this room know someone or like we've interacted with someone at some time that has diabetes. Worldwide, there's more than 180 million people that are suffering from diabetes, and it's one of the fastest growing epidemics worldwide. In the United States alone, I think there's somewhere right now, currently, between 28 and 29 million people suffering from diabetes. There are 2 types of diabetes. There is the insulin-dependent type, which is mainly autoimmune disease that either people find out about when they're children, or later, some people do find out when they're closer to adult, in their adult age. But most of the people that deal with diabetes, 90%, at least in the United States, are type 2 diabetes, which is the most preventable type of disease that you can possibly get. It's actually the only non-infectious disease that the World Health Organization has classified as epidemic. It's completely preventable, completely controllable, and yet it's still one of the fastest growing diseases nationwide or worldwide. And not surprisingly, it's associated with high levels of obesity and physical activity, which is very relevant to us since Philadelphia just got voted our country's most unhealthiest city. So, I mean, what is the real damage of diabetes? So, the problem with diabetes isn't so much the getting of diabetes, it's the fact that when you don't regulate or control your diabetes, it leads to a lot of vascular problems. So, one of the main vascular-related disorders from diabetes is atherosclerosis, which is a building up, if you can see here, you have a normal artery, but as you develop atherosclerosis, you build up these plaques on the inside of your arteries, which tend to narrow your arteries and make blood flow harder, which leads to things like stroke. So when you hear a lot of people have strokes, I mean, a lot of times it's from development of atherosclerosis, and in case of diabetics, that is usually what prompted it, unregulated, uncontrolled diabetes. So it's a huge public health concern. Especially here in the United States, where we don't necessarily have health insurance. So, how does diabetes develop? So, like on a smaller scale, our lab is trying to figure out the mechanisms behind it, or how it works. So, to understand that, you need to understand how your arteries are composed. So, inside your arteries, the cells that line the inside of your arteries are called endothelial cells. They are the major, vasoregulator, the control center of your vasculature. They control things like permeability, vascular tone, which is how your arteries compress or constrict or vasodilate. They control inflammation, angiogenesis, basically anything. A lot of people like to say it starts and ends with endothelial cells. They are the main vasoregulator. And along with that, endothelial cells lie on top of what we know as the basement membrane. So it's like a nanoscale mesh of proteins, which helps to support and stabilize endothelial cells. The interactions of these two are what keep you healthy, and any type of dysfunction of them has been shown to lead to vascular disorder. So we know that there's a direct link between endothelial cell dysfunction and vascular disease development. So in addition to interacting with the blood and other biochemicals that are being secreted, the endothelial cells also have a major mechanoregulatory component. So they also sense the flow of the blood. So your blood flows through your arteries, causing shear stress, which is represented here. And it also causes the actual arteries to expand in response to the blood flowing through them. So that's what we call pressure stretch of your arteries. So these two forces elicit two very different responses when isolated. Shear stress causes your cells to elongate and align in the direction of flow. If you think about drag, so if you're aligning in that direction, you're going to limit the drag on any one cell, which decreases the resistance across the cellular layer. Whereas when you're looking at stretching, cells are actually going to align perpendicular to that flow, or perpendicular to that stretch, which I'll show you here. So my project mainly deals with this pressure stretch, which is also known as strain. So when you have cells in a static condition, we're all just in ambient directions. There's no particular alignment. However, once you add a stretching in a particular direction here in a horizontal plane, the cells begin to elongate and align perpendicular to that stretch. So it's for the same reasons that we see with shear. That overall realignment limits limits the resistance, limits the actual force on any individual cell. And this is just showing the same thing, but looking at the actin network. The actin network is the cytoskeleton, so it's like, for us, it's like our bones. It's what gives the structure to your cell. So again, this is just another way of showing that in response to cyclic strain, you're getting elongation and alignment of your actin network perpendicular to that. And again, any loss in hemodynamic control also leads to endothelial dysfunction, which goes along the pathway to vascular disease. So, like I said before, endothelial cells are supported by this thin nanoscale basement membrane protein network. So they also have they play a large role in controlling how the basement membrane organizes. So here on the top two images, these are endothelial cells taking a much lower magnification that are aligned because they were grown on microgrooves. So they will automatically want to align in whatever direction. So even if you're not applying like a shear flow or strain, they're still going to want to align in the same direction as whatever surface they're on as well. Whereas these cells are just on smooth glass, and they are just randomly dispersed, randomly oriented. Now here we're looking at the basement membrane collagen network underneath these cells. So if you look underneath these using polarized light, so without getting into the intricacies of polarized light, collagen has a natural birefringence. So when the collagen fibers in the basement membrane are aligned, you can use polarized light and you'll see a uniformity of color. Whereas if you're looking at a more disorganized, non-uniform basement membrane, you're going to see this, you're going to have multiple colors shown. It's kind of like if any of you have Oakley glasses, they're like most Oakley glasses, the reason why they're so expensive, have a polarizer in them, and if you actually turn them in the light, you'll see the colors change, and if you have them aligned in certain directions, it'll look more uniform than others, just for a simplified example. So this just shows that, in addition, just endothelial cells, it's important for them to regulate biochemical signaling from proteins and things of that nature, mechanical forces, but it's also important for them to interact with their basement membrane. So any changes in the basement membrane are going to directly affect endothelial cell. So then why is diabetes and uncontrolled hyperglycemia important, critical to this endothelial cell basement membrane unit? So when you have hyperglycemia, you have this reaction that takes place in your basement membrane proteins known as protein glycation. Specifically, when we're looking at collagen, you have collagen glycation, which collagen is the bulk protein in the basement membrane. And this glycation is shown to directly lead to atherosclerosis and other cardiovascular diseases. And I'll show you a picture of the reaction in a minute. But just so you know, the glycation reaction is the same reaction that takes place when you're cooking your food. So that same browning reaction that gives your food, your turkey, your chicken, and just for vegetarians, your Brussels sprouts, so much flavor, is the same, literally the same reaction that's occurring in a patient with diabetes in a hyperglycemic, unregulated environment in their arteries. So this glycation reaction is just a reducing reaction where you take a reducing sugar, in this case glucose, and it reacts with the protein. In our case, we're mainly looking at collagen. And instead of having a nice loose collagen structure, which we know is a triple helical, it's very organized, glycation causes more cross-linking and more covalent bonds to form across the fibrils, which causes it to become more cross-linked. They've shown in small animal studies that their arteries do become stiffer. And in addition, you lose permeability control, changes in adhesion, and vasodilation effects that the endothelial cell is responsible for. The main one of which my thesis focuses on as well is adhesion. So we know from a biochemical standpoint that in hyperglycemia we get this collagen glycation, which leads to impaired endothelial cell function and atherosclerosis. So we get this plaque formation and this narrowing of arteries. And we've also known for decades and decades and decades, people have been studying shear stress or cyclic strain effects on endothelial cells, and we know that as long as this is maintained and the cells adapt, they maintain their atheroprotective nature. They keep, that's how the cell maintains health. It responds to some change in your blood pressure or some change in viscosity of your blood for whatever reason to maintain atheroprotective status. However, we do know that when you lose shear stress and response that you do become atherosclerotic. However, there's been no studies to look at when you look at mechanical forces in a disease state such as diabetes or any other diseases. No one's really looking at what the mechanical effects of disease are on the body. So how do the arteries change in stiffness and then adaptation to different disease states. And specifically for us, we wanted to look at diabetes. So how is the body changing in response to diabetes? So our lab is really big in building devices to use to study different disorders. I'll actually show you a few at the last slide, but this is, so I look at cyclic strain, so we built a very simple uniaxial cyclic strain device, which basically, this is a motor, and it translates, it goes from roto to linear motion to stretch a movable clamp, and on this side there's a fixed clamp, and it stretches elastic membranes. So those elastic membranes we can treat and make them more hydrophilic so that our collagen or our glycated collagen will stick to them, and then we can put cells down on them. And it's like mimicking an artery. Basically, we're mimicking the artery to apply stretch to these cells. And this is how it looks right before. So what did we find? So when we looked at in our static cases, we expected to see that on native or glycated collagen, the cells initially just have a typical cobblestone shape. So the red here is just actin, and the blue are cell nucleus. So there's no general organization to the actin network. However, after 6-hour cyclic strain, we see that the active cytoskeleton on native cells is elongating and aligning, whereas on glycated, we still saw very cobblestone morphology, which is very typical of endothelial cells. We have a software MATLAB code in our lab that we use to analyze the actin fibers, and essentially we found that over time, as you can see in red, on native collagen, we do get the typical alignment of our cells towards a 90-degree perpendicular to the direction of applied strain. However, our glycated collagen, it was completely disrupting reorganization of endothelial cells. And we've actually seen this as well in response to shear flow as well. I'm only showing my data, but we've seen the same recurrence. So essentially, glycated collagen is disrupting endothelial cell response to mechanical forces. And now we really want to know the reason why. I won't take you through all of that because that's like my whole thesis. But this is where we kind of started. We know that on native collagen we have, there's these things called integrants, which are essentially the linker adhesion molecules between the basement membrane protein, in our case collagen, and endothelial cell. And we know that those molecules are directly responsible for controlling adhesion. So we just looked into those, and we found here, skipping ahead, we found here that in response to strain, at 10 minutes, we have, well, it's hard to see, but the purple shows that we have increased focal adhesion activation, whereas on glycated collagen, we didn't see the same activation. And this activation is what downstream from the pathway leads to more focal adhesion formation. So basically, in addition to blocking the actual response to mechanical stretch, the reason we believe it's happening is because glycated collagen is disrupting adhesion to the basement membrane, which is what we have here. So we know that we're getting diminished focal adhesion activity once we switch from native to glycated collagen, and we know that there's increased cross-linking and stiffness of this glycated collagen, which could be impairing binding sites. So now the research from there went on to investigate, is it more of a upstream from this? Is it impairing how cells bind to glycated collagen? Or is it downstream, is that they're binding fine, but they can't turn over their focal adhesions in order to remodel? So again, that's what we found. And well, that's what motivates a lot of the research in our lab. We've lost this adaptation, and we've also lost adhesion formation. So after this is more biology, more chemistry, more protein-protein interactions, and engineering to make more systems to investigate these pathways. I mean, obviously, we're time limited today, but if you want to come to my thesis defense, we can go over all of this in all gory details. But it's very interdisciplinary, which is what I'm trying to show you here. Like, our lab is actually right at the intersection of biology, chemistry, and engineering. And why is this all important? So in addition to just understanding how endothelial cells and basement membranes interact, the bigger thing here is we'll understand atherosclerosis and aging more. These are the same reactions that take place in the collagen in your skin and like the sag that eventually occurs. So, I mean, it has universal applications. In the long term, we want to get better drug delivery. I mean, I don't know if you guys know a lot about the drug industry now, but it's kind of a trial and error situation. It's not so much a targeted, this will target the problem, this will just mediate the symptoms for now. It's kind of where we are with drug development. So again, this is my lab. I work with Dr. Elisa Klein in mechanical engineering. And this is just an overview of everything our lab kind of does. Again, so the shear response, endothelial response to shear stress, to strain, which I showed you. Now we have other devices we're using to look at adhesion. We are now into drug development with nanoparticles, as well as using the natural capacity of the basement membrane to bind and release things to be kind of a natural drug delivery target, as well as other single cell mechanics, which is really interesting, and looking at changes in cell mechanics on different substrates of different substances. So if you have different people with hardened arteries versus softer arteries, how are their cells adapting without the external application of mechanical forces? So we do a lot, and we build a lot of devices to do a lot. That's it. Questions?
Thank you, Dannielle. Our next speaker is Jonathan Soffer from the Department of Chemistry, and he will talk to us about his work.
I'd like to thank the Graduate Student Association as well as the Graduate Studies Office for organizing this event. I think it's great to see all the interdisciplinary science in action. Today I'm going to briefly describe, or rather quickly describe, protein folding and protein frustration. When I was approached, I was first asked about the opportunities that I've had as a graduate student here at Drexel. And I think first and foremost was the opportunity for the PhD itself. I did a real quick search online at the US Census, and apparently this degree is only held by 1.33% of the US population, which I thought was kind of incredible. And if we expand that to all the professional degrees, doctors, this only goes up to about 3%, which is just such a small percentage of the US population. It's an opportunity that just is very, very significant, I think. I'm speaking as a former president of the Graduate Student Association, as well as secretary, and I think that all of this combined led to significant development of my scientific and professional presence through conferences, invited talks, and various committees that I've sat on across the university, from main campus all the way over to Sacramento's campus. My primary research interest is biophysical chemistry and biospectroscopy. The spectroscopies that I'm primarily going to mention in this talk is absorption spectroscopy, electronic circular dichrosum, which I'll refer to as CD later on, Raman and resonance Raman. And my focus is mainly on the structural analysis of proteins in solution, as well as bound to the surface of liposomes. And this is to develop an understanding of the unfolded state of proteins, as well as the protein folding mechanism. So briefly into the classical view of protein folding, proteins are involved in almost every biological process, with a defining characteristic that they have the ability to self-assemble quickly and with precision. So breakthrough study was done by Anandan, illustrating how ribonuclease can reversibly denature and renature in vitro. So that's an unfolding and a refolding by methods of temperature change and pH. And his overall conclusion was that native state information is encoded in the primary amino acid sequence. So if you're looking at a long strand of amino acids, as this thing folds, it has a three-dimensional structure that will also perform a function. So this led to a thermodynamic hypothesis. The native state, which is the fully folded state, is the most thermodynamically stable. And the folding process must therefore be a consequence that drives it to a minimum energy, and that's the minimum free energy, which is delta G. But how does this occur? This led to a series of thought experiments by Leventhal, which is now referred to as Leventhal's paradox. So he assumed that each amino acid oriented itself into three isoenergetic states. So these are all states that have the same energy. And for 100 residues, or the protein that I study is 104 amino acids, or 104 residues, that leads to a possibility of 3 to the 100 possible configurations. So if we think about this a little bit further, if the protein can sample a new configuration every 10 to the minus 13 seconds, it would take 10 to the 27 years in order for this protein to sample all possible configurations. And this is significant because it's longer than the age of the universe. What we know about this, then, is that the proteins fold reliably, reproducibly, and in short time scales. These are in microseconds. If this didn't happen, then none of us would exist. So this then led to proteins must fold in some sort of prearranged way, which was referred to by Anandan as a folded pathway. And as experimental techniques improved, new questions arose from the population of intermediates and how they should be considered. So as these other not natively folded or not fully folded states arose, were these in the pathway, or were they somewhere outside of this unfolded to folded state? So if you're looking at the chain as it's folding, there's both attractive and repulsive interactions between parts of the chains. If you think of it from a random sequence, typically it cannot satisfy all of its structural constraints without bringing some sort of relatively repulsive part in proximity to something else that is in the chain as it's folding. And to best describe these possible deviations in the path can be in terms of a funneled landscape. So this is an example of a funneled landscape that's used to explain this. So in looking in more details now at funneled landscapes, not all conformations are equally likely. The energy surface can be viewed with a variety of local minima. So basically, an unfolded protein is going to go down a pathway into a folded state. And the more rugged a landscape can hold kinetic traps, energy barriers, and narrow throughway paths on the way to the native structure, which will allow for the folding process to be multi-state, and it'll also allow for different times. So this makes a lot more sense when you're looking at different proteins that take different amounts of time to fold, and it kind of brings everything into perspective in the grand scheme of things. Further looking at this, approaching intermediates and misfolded states, as it approaches the native state, the occupation of a misfolded state is not unusual in the folding process. As long as the free energy remains above the glass temperature of the folding funnel, the barrier between the folded and the misfolded can be overcome, and this allows the folded protein to become predominantly populated. What that means is there's going to be energy wells on the way down to the folded pathway, and if it can get over that well, it'll continue down the path. This leads us to protein frustration, which is going to be primarily what I'm going to be speaking about. Even the lowest energy states can have sets of interactions that are frustrated. So these are interactions within the protein that are non-native, but will be a minima in that overall landscape. These states sometimes cannot escape these wells, but they're often difficult to discover spectroscopically. And it's just because of the fact that proteins are going to avoid this as it's continuously going down the landscape. And one of these states is going to be the focus of later on in the talk. So the protein that I study is cytochrome C. It's located in the intermembrane space of the mitochondria, where it's loosely associated to the BC complex. So this is the mitochondria. This is the electron transport pathway. This is the BC complex. This is it blown up. And this is cytochrome C. Cytochrome C is the key mediator in the electron transport chain, where it transfers an electron from complex 3 to complex 4. And it aids in the production of ATP, which is pretty much the energy currency of the cell. Just going quickly into the biological relevance, over the last 5-10 years, cytochrome C's research has shifted into understanding its role in a variety of diseases, the first of which is the apoptotic process. In this, cytochrome C acts as a mediator in triggering this, and it's a programmed cell death. It's also been linked to alpha-synuclein aggregation. This is one of the linkers, or one of the starting points for Parkinson's disease, and it's possibly linked to a variety of other neurodegenerative diseases. Looking at cytochrome C now in more detail, it's been a model system for protein folding studies. It's a small heme protein, about 12.4 kilodaltons. It's highly soluble. As I mentioned previously, it contains 104 amino acids, contains 5 helices, and this heme prosthetic group in the center. And this is located in the hydrophobic region within the protein. Of primary importance for the rest of the talk is what's actually ligated to this heme. So this is the heme. It's covalently linked to the protein through 2 thioether bridges, and these are both cysteines, cysteine 14 and 17. And as the 5th ligand underneath of the porphyrin is a histidine 18. And then something that's pretty unique to cytochrome that enhances the stability of the reduced state is methionine 80, which is the 6th axial ligand in this protein. So now I'm going to go into some details of some recent findings in my lab, which was the population of a misfolded state under folding conditions, which is something that's not regularly seen. So our group used a new modified method for testing cytochrome just to avoid photoreduction of our sample when it was exposed to the laser for Raman experiments. So what this meant was we put cytochrome in solution, we brought it up to a pH of 11, and then allowed it to sit at that pH under oxidized conditions for varying amounts of time. So looking at this first set of data, which was after 2 hours, we saw something very interesting occur. This 695 band that I have marked here is a marker band that tells us if the protein itself sits in its native state. So it's that methionine iron linkage. And something that was new that showed up is this 625 band. So this was something that was never seen in solution before. And as you can see, as the pH decreased, this band began to increase in intensity. So this let us wonder, is it possible to completely remove the population of the natively folded state and allow the population of this misfolded state to become increased. So after about 8 months of experimenting, if we exposed the protein to oxidizing conditions for an extended period of time of a week, that 695 band would completely disappear, and the 625 band became more and more intense. And just to keep in mind, this is a high spin marker band, which is seen in some bacterial cytochrome Cs. From the titration data, which is this here, the PKs told us that it was histidine. So through site-directed mutagenesis, we believe that this histidine 33 is actually coming down below the protein. This methionine 80 is breaking its ligation and becoming axially ligated to the heme. So you still have the histidine 18 intact, and now you have a histidine 33 intact down here. So it's a bishistidine. But this wasn't the end of the story. Another diagnostic that we run is always circular dichroism, and there was something interesting that occurred as well. So this is an overlay of the 1 day and the 1 week spectra. And normal native cytochrome C is this couplet always shows up that's indicative of a methionine iron bond. And for the 1 week spectra, this goes away. And if you remember from the previous slide that I mentioned that from the charge transfer band that it was in a high spin state, this is now telling us that it's in a low spin state as it goes to a positive couplet. So the metal center in the metal in the center of the protein is actually changing its states in the same sample, just by a spectroscopic method. So then, third interesting finding is, I always go to Raman because there's the heme center, it resonates very intensely, and looked at the 1 week sample again. So what was interesting is, I was worried that the protein was in a reduced state, because this is something that regularly occurs because it's more stable. But in the Raman, I saw a marker band that indicates an oxidized state, which was great news to me. But something even more interesting was this band right here, which is indicative of the low spin state again. But it's also around the wave number for something that's called a quantum mixed state. So this is something that's kind of in a flux between the high spin and the low spin state. And this is only seen in bacterial cytochrome Cs previously. So this is a brand new finding of a frustrated state of the protein. So just to kind of bring this all together, we found this very interesting as a possible first example of horse heart cytochrome C populating a partially stable, unfolded state at conditions that normally is a fully folded protein. So this was very, very exciting to us. This illustrated the occupation of a frustrated state in solution, which doesn't usually occur. If it's a membrane protein, it's usually when it's interacting with the surface of that membrane or whatever environment that it's regularly in. The state resembles an off-pathway misligated state of cytochrome C that temporarily occupies upon denatured refolding, similar to that of a misfolded state found on the surface of negatively charged liposomes. So what that means is we're looking at our system in the most simplified form. In a cell, it's much too complicated for us to really figure out what's going on. So we break the system down into pieces, and then we try to build it back up in order to figure out the complexity of the more complicated system piece by piece. So our liposome system, where this is seen, is a very simplified form of the mitochondrial membrane. So this leads us to believe on the outside surface of the protein that positive patches create an alkali misfolded state, which would allow for peroxidase activity. And this peroxidase activity is a requirement for cytochrome C's release from the mitochondria, as well as possibly one of the triggers for the apoptotic pathway. So it was just very, very exciting findings in our group. So hopefully I've illustrated a little bit of a variety of, my group does a lot of chemistry, biology, and physics, as you can see. I cut out a bit of the physics just because of the time restraints, and that's all. This image is 300 overlaid images of the unfolding process of cytochrome C in solution. So, thank you.
Thank you very much, Jonathan. And last but certainly not least is our third speaker from the Department of Civil, Architectural and Environmental Engineering, Michael Ryan.
Good morning, everybody. As was mentioned, my name is Michael Ryan. I'm from the Department of Civil, Architectural and Environmental Engineering. Just a little bit about myself. I am from Jamaica, tiny little island off the south coast of Florida, about 500 miles. An island characterized by its beautiful waterfalls, white sandy coral beaches, some of the fastest people on the planet, and of course, reggae music. Now that I've plugged for tourism, what I do. Overview, I'm going to be doing some of the motivation behind the research that I do here. A brief synopsis or look at microbial source tracking, what it's all about. Some of the application and the conclusions that have been made so far from the research we have done. So, basically the motivation is we're looking at water systems, the environmental watersheds. And what happens is, unfortunately, when you do tests in a lab, those results does not translate to complex environmental systems. So primarily I'm trying to determine what are the sources of microbial pollution in your watershed. So we could have pollution from your farms, industrial complexes, your wastewater sewage treatment plant. The question is, the problem is, when these water bodies are polluted, we are not sure where they're coming from. Regulatory utilities need to know exactly, do we need to fix the water treatment plant, or is it the farm upstream that's causing the problem? So overall, my primary focus is on water. We're looking at water consumption and recreational activities. And of course, there's pathogens lurking everywhere in our water systems, ready to get you ill, as Natalie can actually tell you a little about. So there are different pathways, as I mentioned, that you can get ill. There are, as I say, human pollution, your farms, or even wildlife. There are different transmission pathways. You have air, soil, water, direct contact. And of course, you get in by drinking the water, touching other people, eating the food, whatever it might be. So there are many ways where infected water can lead to you getting sick. So now, not to complicate the matters, there are several different types of pathogens that can get you sick. All of them interact in different ways, giving different symptoms. All of these that are listed here are primarily transmitted by water. You're not sure which one it is. The problem is, can you actually test and identify each one? It's not economically feasible unless you know something about the symptoms. You can't really say, okay, go test for, say for argument's sake, salmonella, if you don't realize that salmonella might be the one causing the problem. It don't make sense testing for 10 different pathogens when you're looking for only one of them. So that's where traditional methodology comes into place. We use fecal indicator bacteria. It's normally a cultivation method. We're looking for an organism that, the assumption here is that these organisms are going to be in the water body or whatever food or whatever you're looking for in more numbers than your pathogens. Their fate and transport in environmental systems are going to be similar to pathogens, or maybe survive a little bit longer. And they are pretty much indicative of enteric, or should I say human intestinal or animal intestinal sources. But there are limitations. One, yes, we say, okay, these organisms are indicating pollution, but where is the pollution coming from? We have found that it's actually acknowledged that 99% of the bacteria that is currently known, we don't know them. We can't cultivate them. And in fact, new technology is even showing that we even don't know what is out there. If without, and one of the technology I'm using, without that, which is called polymerase chain reaction, without that technology, we would not even know that some of these organisms exist. We have cultivation biases. Yes, we have a media that you grow one particular organism, another one is not going to grow on it. So how do you correlate what's going on? They are not representative of all pathogens, especially viruses. Yes, say for example, the fecal indicator that is currently used for traditional measures of pollution is your coliforms or your E. coli, they do not necessarily represent viruses or how viruses react in the environment. And to make matters worse, we have now found that some of these organisms actually grow in the environment. So there's these complex scenarios that we need to actually figure out, especially when it comes to environmental samples. Now, the question or main focus behind my research is what exactly is causing the pollution? Is it animals or is it human? If it is animal, what type of animal? So we are looking at the assumptions that there is a traceable association with microbes that are, should I say, isolated from the intestinal structure of animals and humans. They may have co-evolved over the years. They may have developed or adapted to the environmental conditions where they grow in. So the argument here is if we take an organism from human, you're unlikely to get that one from, say, dog. If you do it from dog, you are going to be unlikely to get it from, say, a deer or a cow. So what we're doing is, in order to do this process, we're doing molecular biology. So not only were we cultivating them, we're doing DNA extractions, which what we're doing is we're taking the cells of the organisms, we're splitting them open, and we're taking their DNA out. Now the problem is we do not want all the information of the entire genome. For example, the organism I'm looking at, it's comprised of 5.5 million base pairs. I'm trying to target one little segment which is right down to 160 base pairs. So I'm not really interested in all of that. The problem is to try and do this type of experiment. This little animation will actually show you what I'm talking about where PCR is concerned, in particular real-time PCR. So we're taking the DNA from all these different sources, we're extracting them, we are putting them in these little tiny tubes, we mix it with all the reagents needed for you to build the DNA. We're trying to simulate DNA amplification in the cells. And we are amplifying them by regulating temperatures, the ingredients we're putting in, and we're doing DNA amplification. One of the unique things I'm trying to do is also incorporate quantification in here. So not only am I trying to make a lot more because we can't figure out what's going with just one strand of DNA. We need a lot. We're trying to quantify them. And what we do is repeat this cycle over and over, and as is indicated there, after about 35 cycles, which right now my process is done in about half an hour, we get maybe 30 billion cells in there. I'm also incorporating a probe to make my process a whole lot more specific. And the idea is I'm trying to target a very, very specific region of the DNA. And of course, remember, we're talking from 5.5 billion million all the way down to only 160 base pairs. And what we're doing, of course, is that the equipment, that probe that we incorporate lets off a fluorescent light where the equipment is able to generate. And after each cycle, we are able to quantify how much DNA it was in our original sample. Now, what we also do is we use gel electrophoresis, which we can also do I use it as a kind of QCA method to determine if what I'm looking for is at the right size. Because you might get amplification coming off, the equipment says it's there, but when you do gel electrophoresis, it's not coming off at where it's supposed to. It comes off at a different level and you know something is wrong. Now when all of these problems come into play, what we have to do is, as I mentioned before, the primary critical part of our process is the primer and the probes. If they're not specific for what you're looking for, you're not going to find what you're looking for. Everything might amplify when it's telling you, okay, this source is human, when in truth and in fact it is not. So that's where a lot of DNA cloning and sequencing come in. Imagine a water drop have all these different types of pathogens lurking. You need to try and identify maybe just one of them. So your probe and primer needs to be extremely specific. So what we will do is take all of these DNA, we do some DNA cloning, which is basically manipulating our vector is normally E. coli. You're manipulating the plasmid, another genetic structure of the E. coli, incorporating the DNA you have just produced with your PCR into them, grow them, and then you send it out for DNA sequencing. And that's just where the fun begins. Because now you have to do bioinformatics, and I won't go into detail, where you're trying to design primers and probes that is going to be specific for what you're looking for, and at the same time not target or anneal to sequences that is going to basically give you false results. And this is actually one set of primer and probe that was actually designed in the research, and is showing that the 4 sequences in here that was associated with humans, associated organisms, were targeted, and the others associated with other animals were not. So, application perspective. We have looked at a number of environmental water bodies. This is just one of them where, for example, this particular stream is located in Philadelphia. The headwater originates here. It's on a farm. And when you use traditional cultivation methods for Enterococcus or E. coli, they go off the chart. It looks almost like it's wastewater. But now there are no wastewater sources on this particular site. When we applied the protocol, we were able to not only detect that there were no wastewater, interestingly we found, okay, this is impacted by human sources, so what could be the problem? So happens that there are CSOs, and those of you in environmental engineering, these are combined sewage overflows that predominates throughout Philadelphia, where this particular one, this was after a rain event, showed elevated levels of human fecal contamination in that stream. And as I said, this was basically proving the assumption that it's worked, although the regular traditional protocol showed fecal pollution, we were able to differentiate and say, no, it's not human, it's associated with animal. And now I'm looking at, okay, can I actually identify what type of animal is actually impacting on these water bodies? We have looked at several other sources, and I won't go into the results, especially because I have to get permission before I can divulge, but we've looked at several different sites of concern in and around Philadelphia and even Puerto Rico, and we have gotten some very, very, very interesting results. Some is confirming what is being assumed to going on. Some is raising more questions as to, okay, exactly what is happening here. And in conclusion, what we have done so far is we've developed, at least here is the human-specific genetic markers for MST protocol. We have actually improved on the assay where we have gotten this, between collection of sample and actual tell you your result, I can give you this in approximately 6 hours. Traditional technology, or should I say traditional cultivation methods, it's like 24 hours for a presumptive, and then you have to run additional tests, and that's like 48 hours. And then of course, if you're looking for pathogens, this might be a couple days. Here as well, we have actually incorporated some amount of quantification into the protocol so we can quantify the DNA, make extrapolation to how much organisms were in there, and overall do risk assessment. So what we are able to do is assess utilities and the regulatory bodies, utilities like PWD who funds this project, regulatory bodies like your EPA, your CDCs, as to what's actually happening, what do you need to do, exactly which source do you need to target to make sure that your water bodies and by extrapolation foods, if you're applying it to that, is contaminated or polluted. I won't go into all the nuts and bolts that goes into all the calculations and all the different aspects of the research. And of course, I have my defense in a couple of months, so if you want more details, please feel free to attend. And now that I've bored you all to death, any questions?
Thank you, Michael. So now we're going to come to the fun part of today's conversations, and that is I'm going to open it up to the floor for any questions for the panelists or in general, some questions about multidisciplinary research for the panelists, for me, for any one of us. Yeah?
I have a question for you.
Sure.
You showed us this funnel, this energy landscape. So obviously the energy is on that vertical axis.
Yeah.
But what are the other axes that are normal in that plane?
The other axes that are normal. So you're asking these, or so this right here is the free energy landscape. Here would be, depending on what system you're looking at, it would be the possible pathway that it could go through as it goes down. I guess the easiest way to think of it would be it would just be delta. It would be a free jibs energy. It's as it's just kind of sampling across. Does that answer? It's just an energy calculation.
So if there are no more specific questions for each of the presenters on their work, I'm going to throw a question into the pool for everybody to think about. What do you think are some of the positive sides of doing multidisciplinary work?
Usually you can see things from a different angle. So like, whereas one you're thinking, say if you're looking at the DNA, like you see it from one angle, like I take it apart this way, it's a very ordered, this is how I do it. But if someone else comes in, it's like, hey, actually this would be more efficient from my own experiences, like in electrical engineering or something like that. You can say like something, this, from my experience, is more efficient. Like it's just viewing it from as I said, it's an angle.
Alice, what do you think?
I mean, I'd say so we have a lot of, we were talking about this earlier this week, and we've solved, I think you mentioned, a lot of the simple things. A lot of the simple problems, I mean, we don't call anything simple, but we've solved a lot of the simple problems across multiple fields. But now we have these complex, non-linear, especially for biology, and we know proteins and everything. These are non-linear like decisions. There isn't just like one way to think about it. And I mean, that's kind of the advantage now for multiple